Today, Doctor, we are from Group 9 with the members of Arvin, Aiman, Hazik, Alia, and Minjun. We'd like to present our project proposal. First, I'd like to present about the clinical issue. Vital signs refer to the measurements of the body's most basic functions. There are several main vital signs, such as respiratory rate, blood pressure, heart rate, weight, and temperature. These vital signs should be measured routinely, as these vital signs will provide critical information about our bodies. For example, heart rate. The reading of our heart rate should always within the normal range. Being too fast or too slow might indicate there's a risk of cardiovascular disease or other medical problems. We could detect the underlying health problems when the vital sign readings are not within the normal range. Apart from that, these vital signs also will prevent misdiagnosis when the doctor is assessing the patient's health. When we visited each department of UNMC, we found that these vital signs play an important role in examining the patients, and the patient's vital sign will be determined before seeking consultation from a doctor. However, measuring these vital signs requires specific medical devices, such as measuring blood pressure, requiring a sphygmomanometer, and measuring heart rate made through ECG. These medical devices are rather costly, and it might not be affordable by everyone. Not to mention, these medical devices usually can only be found in medical institutions. Thus, to ensure everyone can have the medical device to measure their vital signs, not only in medical institutions, but could measure it anytime and anywhere, we like to suggest a smartwatch with these features to take over the medical device's role. Unfortunately, not all current smartwatches have been certified as medical devices due to their inaccuracy. Therefore, we have to test the accuracy of the current smartwatch. With it, the smartwatch will be rather more affordable and has a function of testing pulse rate, SpO2 level, and ECG, will be able to provide a more reliable vital signs reading and can achieve our aim, where people can monitor their vital signs anytime and anywhere. Now, I will pass the floor to Alia to discuss about hypothesis. Thank you.
Hi, I am Ali Irene, so I will be explaining on the hypothesis of our project proposal. So for this project, we have proposed 3 hypotheses where it is an assumption of the outcome for the 3 things that we are testing in this project. So therefore, from this experiment, we will test whether the hypothesis can be accepted or rejected. The first hypothesis that we propose is the reading on the pulse oximeter are much more accurate compared to the reading that is shown in the smartwatch for pulse. The second hypothesis that we propose is the reading in the near-infrared spectroscopy, which is the NRIS device, are much more accurate compared to the reading shown in the smartwatch band for SpO2 level. The third and last hypothesis that we propose is the reading on the ECG monitor device are more accurate than the readings shown on the smartwatch band for ECG.
Hello, my name is Muhammad Manibin Sunde. So now I'll be presenting about our lab 1, which is the pulse measurement test, fingertip oximeter versus a smartwatch. So some background information about fingertip oximeters. So they are able to measure pulse and oxygen saturation levels by using lab absorption theory. So how they work is they clip it onto a patient's finger, as seen in the diagram there, in the figure there, and they are compact and portable and non-invasive. Everyone of all ages can use it because it is painless and tolerable. Even though you clipped it onto your finger, it is very tolerable levels of pain, and the results can be obtained in a very short amount of time. It will display 2 numbers, which are the blood oxygen saturation level and your pulse. But in this lab, we are only using this oximeter just to record our pulse measurements only. So now I'll be talking about the mechanism of fingertip oximeter. So it uses photoplethysmography, which is used to detect blood volume changes. PPG is also known as a pulse oximeter waveform. So there are 2 types of PPG, which are transmissive and reflectance. The fingertip oximeter uses the transmissive PPG method, which is when the LED and the detector are opposite of each other, as in the figure in A right there. So it uses infrared light, which has a 900 nanometer wavelength, and laser red light of 640 nanometers wavelength, which will then be projected to a photodetector at the other side of the finger, like in the figure there. So then the light intensity from the source to the detector will depend on the volume of the blood in the arteries. And as the volume of the blood changes with the cardiac cycle, the light intensity also changes with the cardiac rhythm, which will then can be employed to extract pulse measurements. So the apparatus and materials or the biomedical imaging components consist of two things, which is the smartwatch and the fingertip oximeter. So moving on to the proposed methodology for our lab 1 here. Subject will wear smartwatch on their right wrist and fingertip oximeter will be clipped onto their right finger. And ensure for each measurement, make sure that the subject is as stationary as possible because movement during measurement will affect the final readings. Then we shall first take 3 readings when subject is at rest. Next, we will move on to a moderate intensity activity, which is walking. Take 1 reading after subject has walked for 1 minute and repeat until 3 readings are obtained. Make sure that the subject has stopped moving and take their measurement while they have stopped moving. Then moving on to a more intense activity, we will do some jumping jacks as shown in the figure there. Take 1 reading after subject has done 30 jumping jacks and repeat until 3 readings are obtained. Then we will record and tabulate all measurements, and the measurements between the oximeter and smartwatch will be compared to determine the accuracy. So the expected outcome is the fingertip oximeter should be more accurate than the smartwatch. This is because the smartwatch uses refractive PPG, in which the photodetector and the LED are on the same side. Reflective PPG is more sensitive to ambient light sources and pressure and has a lower signal to light ratio as compared to transmissive PPG, which is the fingertip oximeter. Next is the smartwatch might displace a little bit after doing activities such as the jumping jack, in which it may displace the smartwatch and affect the final readings. So that is all from me. Thank you.
Good day, I'm Arvind Pillai and I'm here to present on the second experiment, which is the SpO2 measurement test involving the near-infrared spectroscopy and smartwatch. Beginning with a brief introduction on the background, NIRS is actually a non-invasive optical method, so it assesses the tissue oxygenation and hemodynamics in a variety of tissues. So in this experiment, we actually calculate the percentage of primary chromophore spectra, which are the oxyhemoglobin and deoxyhemoglobin, as well as secondary chromophore spectra, which are the oxymyoglobin and deoxymyoglobin. Then the total blood volume in the tissue is calculated as the sum of O2Hb and HHB. The NIRS are based on 2 properties of human tissue as stated in the slide, and then the advantages of this method is it's feasible to monitor non-invasively, affordable, and then constantly accessible, as well as it does not need any specialized infrastructure as everything they need comes in a kit, and then it does not need specialized staff as the manual is sufficient enough for us to use on our own. Theoretically, the Lambert-Beer law is used to compute the absorption of chromophore in non-scattering clear material. However, as we are using the biological tissue, a scattering medium, in order to increase the optical path length, DPF is included into the equation. So the original equation, which is one on the left, is then converted into the one on the right, together with the description as below. As you can see in the diagram below, the device consists of a transmitter and receiver positioned on the identical plane of the probe. Then, a near-infrared light source with 2 wavelengths will be employed. The distance between the light source and reception sensors determines the depth measuring range from the surface of the skin. As the device shown in the previous diagram, we are using the Potamon, a wearable wireless near-infrared spectroscopy device, to measure the muscle oxygenation. Along with it, a software called Oxysoft is used for data analysis. The data is analyzed within Oxysoft using certain typical analytical methods. Then the data is exported to conventional data formats, and MATLAB function called Oxysoft to MATLAB is used to allow the data to be immediately exported. Parameters and materials used in this experiment are listed as below. So for the methodology, it's broken into 2 stages. The first one involves the setup. The subject wears the smartwatch on the right wrist, and then the Oxysoft software is installed and run on the laptop. The Potamon is turned on and its battery status is checked, and then it's connected to the laptop via Bluetooth. Then the Potamon is covered by plastic wrap, vacuumizing it for sweat protection. Then the Potamon is placed onto the dorsal side of the left wrist, and then covered and wrapped by using a black cloth, and then continued by wrapping with another piece of cloth as well as an elastic strap to secure it in position during the experiment. In the execution stage, the recording is started at the software, then the subject undergoes the first activity, which is resting for a minute. Once the activity is completed, the recording is stopped. The SpO2 level of the subject is measured immediately in the smartwatch as well. Then this particular activity is repeated again 2 more times, and then the stage is repeated by replacing the activity with a minute of walking and 30 seconds of 30 jumping jacks performed by the subject. And then the data obtained at the Oxysoft software is downloaded, all results are recorded and tabulated. The data obtained from the smartwatch is compared to the data obtained from the Oxysurf software. Then the effectiveness of the different devices measuring SpO2 level can be reduced. For the expected outcomes, based on the hypothesis, the readings obtained from the NIRS device should be more accurate compared to the ones obtained from the smartwatch for the SpO2 level, so that as the analysis of the smartwatch can only show the SpO2 level of a single value once the activity has been completed. It is actually limited to a single point analysis. The analysis obtained from the NIRS actually consists from the entire duration of the activity and has a continuous assessment data. So the data obtained from the NIRS has a more precise method of analysis in comparison to the data obtained from the smartwatch via reflected green light PPG. That's all for experiment 2. Thank you.
Hello everyone, my name is Hazik and I'll be continuing on our lab 3. Our lab 3 is in regards to the electrocardiogram, where we'll be comparing the ECG of a smartwatch and the ECG of a ECG machine. Okay, now before I go further and talk about the whole lab itself, I'll give you a bit short of an intro or background onto what ECG is. Now ECG stands for electrocardiography, or also known as electrocardiogram. Now what this is a non-invasive electrical signal method to determine the heart's electrical activity. Now this is done through the skin, as you can see from the diagram on the bottom side, where the patient is and the electrode is placed on the skin. And then the signal can be seen at the here. And as you can see, it appears to be like a series of waves, and there's a peak there also, and they come together to form one ECG waveform, as you can see at this point. Now this waveform is consisting of the P, Q, R, S, T waves, and this wave each has a meaning. Now the P wave is where there is an atrial depolarization of the atrium, the top half of the heart, and the QRS complex represents the ventricular depolarization, and the T wave means ventricular repolarization. What is the use of this ECG? Why do we want to look at the electrical signals of the heart? And it's actually quite significant in identifying the patient's cardiovascular health. One of the ways that is significant is that a clinician would actually calculate the time intervals of each wave and intervals. So as you can see, there's the segments, there's the intervals, and a clinician will calculate the time it takes for each one of them. And from there also, a clinician can also, or a doctor can look if it has an abnormal shape of the ECG waveform. And this could mean atrial or ventricular fibrillation. It's where the heartbeat is not synchronous. Furthermore, it can also be used to achieve a heart rate reading. This is what we'll be looking at in this lab. This can be done by calculating how many R waves there are in a minute, or there's a shorter way to do it, which is calculating the interval between two R peaks. So how much time it takes between two R peaks, you can calculate how many beats there are per minute. Now for this lab, what we are proposing is that we are proposing a comparison, because some high-end smartwatches, they have an ECG functionality. Most of the low-end smartwatches don't have an ECG functionality. We'll be only looking at the high-end smartwatches. Now we're going to test the difference between the two to see which one is more accurate. And in order to do that, we're not going to be comparing the waveform or anything. We're going to be looking specifically at just heart rate of the two devices. Now onto the apparatus and materials that we'll be using in this lab, lab 3. There's going to be disposable ECG electrodes, loop prep skin prep gel, gauze pads, nitrile gloves, ECG machine, bicycle ergometer, a cotton, and also a smartwatch. Moving on to the methodology, we will start with the preparation for the lab. Now we will propose that in order to start, we will start by laying the test down on the subject, laying the test subject on the bench, and then we will identify the points for electrode placement. Now the points that we'll be using is according to this diagram. As you can see, there's RA, LA, V1, V2, V3 until V6, RL, and LL. The points will then be cleaned using the loop prep gel and the gauze pad as mentioned before, and the electrodes will be placed on the points. And then it doesn't matter which electrode that we place, just place it. And then when we are done with that, we'll connect each of the electrodes with the corresponding cables from the ECG machine. Now that we've prepared the subject, we'll be continuing on to the data collection. In order to collect the data, we start with the resting heart rate. In order for us to do that, we ensure that the subject is in a rested state, so we ensure that the subject is not like after a jog or walking to the lab. We make sure that he's rested and he's breathing normally. After that, we record the ECG of the subject using both devices. So each of the recordings will take 1 minute, we'll let him rest for 1 minute, and then we would take the heart rate of the ECG, and we will also record the heart rate of the smartwatch. After that, we would have the subject sit on the bicycle ergometer, and this is almost similar to before, but then we have to start with 1 minute of the subject cycling with a load of 50 watts, if I'm not mistaken. And this load is one which the subject mentions to be similar to walking. And then we will walk for 1 minute, and then we will record the ECG of the subject using both devices once again, and record it 3 times. We will record it, and then we will start with cycling, and then record it, and record again. Then the subject cycles with a higher load of 100, which simulates somewhat of a running situation. And once again, we will record the ECG using both devices. And once we record, we start over with the running, with the cycling, and then we continue with recording, and then back to it again to get 3 readings. Once we finish with that, we tabulate the heart rates and compare. And what do we expect out of this lab? Now, for now, our guess is that the ECG machine will have a higher accuracy of a heart rate as compared to the eSmartWatch ECG. Now, the reason for this is being that whenever the patient has to record the ECG, the patient has to hold on to it, like has to like, touch it. So I was thinking, I'm assuming that the reason why that is because it has to read the ECG, the electrical signals from the finger at the same time also on the wrist. And as compared to the ECG machine, which actually has proper electrodes other than just touching, proper electrodes that are stuck onto the skin at many points from V1 all the way to V6. And these different points actually give it much more of an accurate reading because more points means they can compare between each of the signals to have a much more cleaner ECG signal. And that's all from me. Thank you.